Hi students, welcome back to the second video module of our NEET 2025 Rapid Revision series, and today's this topic of discussion will be molecular basis of inheritance. So as you know कि मैंने बोला था कि हम पहले जेनेटिक्स वाला पोर्शन कवर अप करेंगे बिकॉज वहां से बहुत सारे क्वेश्चंस आते हैं. लास्ट 6 years में इस वाले चैप्टर से अराउंड 30 क्वेश्चंस आए थे. That gives an average of 5 questions per year and that gives us around 20 marks. Okay. So let's not waste our time. You can visit our website impetuslearning.in for more details and register and book a one-to-one personalized free demo class. Okay. So now we are moving to the chapter that is molecular basis of inheritance. You know it is a very big chapter. So I have divided the chapter into 6 videos and that will be under the playlist of molecular basis of inheritance. So the first video module like lecture number 2A. So that is on the the DNA, the structure of DNA. And the discussion will be as continued as per the syllabus. Okay. So जैसे हर वीडियो में मैं बोलता हूँ कि आई हैव मार्क द एनसीईआरटी फॉर यू गाइस, ताकि तुमको एग्जाम के पहले रिवीजन करने में थोड़ी आसानी हो. कौन से लाइंस तुमको ऑब्वियस रिवीजन करने हैं, कहाँ से क्वेश्चंस आ रहे हैं, कहाँ से क्वेश्चंस आ सकते हैं. आई हैव मार्क्ड ऑल दोस फॉर यू. एट लास्ट वी हैव kept some PYQs. ऑब्वियसली सब पॉसिबल नहीं है. We will keep in those in our DPPs. Okay, so you can get our DPPs by contacting us on this number. Okay, on this number. Okay, तो शुरू करते हैं. Okay, so basically हम जानते हैं कि deoxyribonucleic acid, that is DNA, right? That is DNA and the ribonucleic acid RNA. They are the two types of nucleic acids found in living systems. Okay, so DNA acts as the genetic material in the most of the organisms, as we know, and RNA, though it is also act as a genetic material, they मोस्टली फंक्शन एज मैसेंजर्स. वो किन में जेनेटिक मटेरियल होता है? मेनली वायरसेस में. ओके. एंड आरएनए में हम दो तरह के आरएनए देख सकते हैं. दैट इज वन टीआरएनए, एमआरएनए, एंड अनदर आरएनए इज देयर, दैट इज आरआरएनए. ओके. सो द प्रोसेस ऑफ रेप्लिकेशन, transcription and translation is basically the main motto of the chapter. So what is replication? Production of DNA from a DNA. Okay, that is called replication. Okay, so what is transcription? That is a production of RNA from a DNA. Okay, that is transcription. And finally, what is translation? That is your production of amino acids or the proteins from the RNA molecules. Okay, now moving on to the structure of DNA. Okay, basically DNA is a long polymer of a deoxyribonucleotides. Okay, the length of DNA is the usually defined as the number of nucleotides. Okay, so the number of nucleotides in a DNA, that is the length of the DNA. Okay, a bacteriophage known as 5174, that has 5386 nucleotides. Okay, this has been asked many times in your NEET exams कि 5x174 बैक्टीरियोफाज में कितने न्यूक्लियोटाइड्स होते हैं. योर आंसर विल बी 5386, दैट राउंड्स ऑफ 5400. ओके, देन दे हैव सेड कि बैक्टीरियोफाज लैम्डा हैज 48502 बेस पेयर्स. Escherichia coli has the 4.6 into 10 to the power 6 base pairs, and haploid content of human DNA is 3.3 into 10 to the power 9 base pairs. So यहाँ के सारे न्यूमेरिकल वैल्यू बहुत ही इंपॉर्टेंट है, वो कभी न कभी नीट में पूछे गए हैं. एंड आई विल स्ट्रिक्टली कंसिडर कि तुम लोग ये वाला पोर्शन याद कर लो. आई एम नॉट अ बिग फैन ऑफ रिमेम्बरिंग नंबर्स एंड फैक्ट्स. ओके, बट ये वाले पोर्शन से एनटी ने बहुत बार सवाल पूछे हैं. एक ट्रिकी क्वेश्चन आता है कि डिप्लॉयड कंटेंट ऑफ ह्यूमन डीएनए इज, तुम गलती से भी ये मत लिख देना. ओके, दिस इज एन इक्वल्स टू 3.3. ओके, तो 2n विल बी 3.3 into 2, that is 6.6 into 10 to the power 9 base pairs. Okay, that is your diploid content. Not your haploid content is 3.3 into 10 to the power 9 base pairs. Okay, so how is the structure of a DNA? Basically DNA has 3 main parts. Okay, that is a nitrogenous base, a pentose sugar that can be either ribose in case of RNA and deoxyribose in case of DNA, and a phosphate group. Okay, so basically there are two terms you need to know, that is one nucleoside, another one is nucleotide. What is a nucleoside? Nucleoside is basically the nitrogen base plus the pentose sugar, that we call is a nucleoside. But when we add up the phosphate, then nucleoside turns up into nucleotide. So basically nucleotide is nucleoside plus phosphate base. Okay, nitrogen base are two types. One is purine, another one is pyrimidine. Okay, purines are 2 ring structures and pyrimidines are 1 ring structures. So basically what are the examples of purine? That is adenine and guanine, and pyrimidines are cytosine, uracil and thymine. These are very basic terminologies and very basic concepts. तुम लोग ये क्लास 10th से पढ़ते आ रहे हो and just I am brushing up your concepts. Okay. RNA में क्या होता है कि थाइमिन की जगह यूरासिल होता है. ओके. Nitrogenous base is linked to the hydroxyl of 1 carbon pentose sugar through an N-glycosidic linkage. They have asked many times about the N-glycosidic linkage question कि कौन से linkage से जुड़ा होता है. Okay, they have given the options phosphodiester linkage, disulfide linkage, N-glycosidic linkage. So your answer will be N-glycosidic linkage. Such as adenosine, deoxyadenosine, guanosine, deoxyguanosine, cytidine and deoxycytidine, and uridine and deoxythymidine. So what are they? Basically, they are the nucleoside and nucleotide. Okay. In DNA, they are deoxyadenosine. In RNA, they are adenosine. If we add up the phosphate, that will form adenine. Okay. Deoxyguanosine, in RNA it is guanosine, and if we add up phosphate, that will give us guanine. Deoxythymidine, uridine, and that will give us thymine and uracil as nucleotide. Okay. So a phosphate group is linked to the hydroxyl of the 5' carbon of a nucleoside through a phosphodiester linkage. Okay. Corresponding nucleotide or deoxynucleotide depending upon the type of the sugars present. Okay. So phosphate moiety attaches to the 5' end of the sugar. तो 5' end में क्या रहता है? phosphate रहता है. और 3' end में क्या रहता है? हाइड्रोक्सिल रहता है. So basically 5' to 3' DNA is basically 5' one is phosphate and 3' one is hydroxyl. Okay, remember the fact. This question has been asked many times. What is the another name of thymine? That is 5-methyl uracil. That is 5-methyl uracil. Okay, that is another name of thymine. In 1953, James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkins and Rosalind Franklin, proposed a very simple but famous double helix model for the structure of DNA. Okay, this question has been asked many times. That's the name of the scientist. Okay, so basically there was a scientist. His name was Erwin Chargaff. So what did he say? He said the amount of purine will be equal to the amount of pyrimidine, and one purine will bond with another pyrimidine. Okay, so basically adenine plus guanine will be equal to the thymine plus cytosine, and adenine and thymine will be joined by 2 hydrogen bonds, and guanine and cytosine will be joined via 3 hydrogen bonds. Remember the fact. This has been asked many times. So what are the double helix feature of the DNA? It is made up of 2 polynucleotide chains where the backbone is constituted by the sugar phosphate. So basically the backbone of the DNA is constituted by the sugar and the nitrogenous base. Okay. They have 2 chains of anti-parallel polarity. What is anti-parallel polarity? If they are the 2 strands, okay, one will be 3', 5', another will be 5', 3'. Okay. बेसिकली क्या होता है कि अगर जिस एंड में एक स्ट्रैंड का हाइड्रोक्सिल ग्रुप रहेगा उसी स्ट्रैंड में दूसरे स्ट्रैंड का फॉस्फेट ग्रुप रहेगा. ओके. Adenine forms 2 hydrogen bonds with thymine and opposite strand vice versa. Similarly, guanine is bonded with cytosine with a 3 hydrogen bonds. Okay, remember the fact. The 2 chains are coiled in a right-handed fashion. Okay, this is called the ZDNA. Okay, this is ZDNA. It is right-handed fashion. Okay, it is ZDNA. And the pitch of the helix is 3.4 nanometers. This has been asked many times. The pitch of the helix. The pitch of helix. Okay, pitch of helix is 3.4 nanometers. Okay, and there are roughly 10 base pairs in each turn. Okay, so basically the distance between the consecutive base pair of helix approximately 0.34 nanometers. Okay, that is 0.34 nanometers. This is the central dogma. This was proposed by Francis Crick. This has been asked many times in the NEET. Who proposed central dogma? What is central dogma? That is the formation of DNA, then RNA, then protein. That is central dogma. So from DNA to DNA, it is called as replication. From DNA to mRNA, that is called transcription. From mRNA to protein, that is called translation. Okay, remember this is central dogma. Okay, now packing of DNA helix. In eukaryotes, we have a proper nucleus, right? But in prokaryotes, they don't have the nucleus. तो उनका डीएनए ऐसे वैसे तो वहां घूमते नहीं रहेगा, राइट? वो भी एक सही जगह पे रहेंगे, राइट? तो तब क्या होता है कि there is a term called nucleared. Okay, in prokaryotes such as E. coli, though they don't have a defined nucleus, DNA is not scattered throughout the cell. DNA being negatively charged is held some proteins that have positive charge. That is called as nucleared. Okay, so in eukaryotes, the organization is much more complex. There is a set of positively charged basic proteins called histones. So basically, this is a histone octamer. Why octamer? There are 8 subunits. There are 8 subunits. Okay. What are they? That is H1A, H2B, H3 and H4 into 2. That means H1A 2 होंगे, H2B 2 होंगे, H3 2 होंगे, H4 2 होंगे. Okay. And the DNA will be coiled around this in 1.75 turns. Okay. That means 1 full 3/4 turns. Okay. Basically, histones are rich in basic amino acids and they are mainly residues of lysine and arginine. This has been asked many times कि histone protein के components क्या क्या होते हैं. Okay. Nucleosomes in chromatin are seen as beads on the string structure. Okay. So they have said कि if we see the beads on the string structure, what is this? That is your nucleosomes. Okay. Nucleosomes in chromatins. Okay. So this has been asked many times. Okay. In non-histone protein, there are 2 types of chromatin. That is one euchromatin, another is heterochromatin. Euchromatin that stains very light and it is loosely packed, so it is functional portion. Okay, and the heterochromatin that is densely packed and stains dark are called the heterochromatin. They are the non-functional part. Okay. So how we found that the DNA was the genetic material? Okay, there are transforming principle that was done by Frederick Griffith. Okay, that was done by Frederick Griffith. So what did Griffith do? Griffith did a great thing. Okay, it was his transformation principle. What did he do? He took a Streptococcus pneumoniae bacteria. Streptococcus pneumoniae. Okay, it has 2 strains. It has 2 strains. That is, one is smooth strain and one other one is rough strain. Okay, so basically, when he injected the smooth strain into the mice died because the smooth strain was virulent, and the rough strain, it was non-virulent. So the mouse survived. Okay, the mouse survived. Okay, okay, the mouse survived. Okay. Then when he heated and killed the S strain, then it injected into the mice lived. Okay. जब उन्होंने हीट किया S वाले स्ट्रेन को, तब क्या हुआ कि माइस बच गया. Okay. Then again when he heated and killed the S strain and then a live R strain, उनको मिला के एक माइस में दिया, that the mice died. Okay. The mice died. So this was Griffith's principle. So what was his interpretation? Ki there was something in the R strain that was transforming the genetic material of the S strain. Okay, so this was the transforming principle. But he could not find a decisive answer to his experiment. Okay, so क्या हुआ कि in year 1933, 3 scientists, Avery, McLeod and McCarthy. Okay, so they did the improvisation of the Griffith's test. Okay, तो उन्होंने क्या किया था? They purified the biochemicals like protein, DNA, RNA, that is from the heat killed S cells to see which ones could transform live R cells into the S cells. Okay, they discovered that the DNA alone from the S bacteria caused R bacteria to become transformed. मतलब तुमने प्रोटीन दिया वो नहीं हुआ, तुमने बाकी RNA दिया नहीं हुआ, जब तुमने DNA दिया तब वो S strain virulent हो गया. Okay. They also discovered that the protein digesting enzymes, that proteases, and RNA digesting enzymes, that are RNases, did not affect the transformation. So the transforming substance was not protein or RNA. Okay. So this confirmed that the genetic material was DNA. Okay. The unequivocal proof that the DNA is the genetic material came from the experiments of Alfred Hershey and the Martha Chase. This line has been asked many times. Also after NEET 2019 and also before NEET 2019. Okay, बहुत बार पूछा गया है. The unequivocal proof that DNA is the genetic material, which was done by Hershey and Chase, Alfred Hershey and Martha Chase. Okay, so उन्होंने क्या किया? The bacteria, उन्होंने क्या किया? एक वायरस को बैक्टीरियोफाज से अटैक करवाया. Okay, so क्या किया उन्होंने? They grew up some viruses on a medium that contained radioactive phosphorus and some others medium that contained radioactive sulfur. Okay. So viruses grown in the presence in the radioactive phosphorus contain radioactive DNA, but not a radioactive protein because DNA contains only phosphorus. As I discussed earlier, कि DNA में phosphorus रहता है, sulfur नहीं. Sulfur रहता है cell membrane में. Similarly, viruses grown on the radioactive sulfur contain radioactive protein, but not radioactive DNA because DNA doesn't contain sulfur. Then the radioactive phages were allowed to attach to E. coli bacteria. Then as infection proceeded, the viral coats were removed from the bacteria. By agitating them in a blender, the virus particles were separated from the bacteria by spinning them in a centrifuge. Okay, तो उनको हमने centrifuge में spin किया. फिर हमने क्या देखा कि bacteria which was infected with virus that had a radioactive DNA was radioactive, indicating that the DNA was the material that passed from the virus to the bacteria. So जिनमें radioactive phosphorus था वो अगले generation में that transmitted. Okay, so वी कैन सी लाइक द यहाँ से कि जेनेटिक मटेरियल डीएनए होता है, प्रोटीन नहीं. ओके. This indicates that proteins did not enter the bacteria from the viruses. Okay, DNA is therefore the genetic material that passed from the virus to the bacteria. Okay. So what are the properties of a genetic material? Okay, what are the properties? It should be able to generate its replica, that is replication. It should be stable. It should allow mutations, and it should be able to express itself in form of Mendelian characters. So this was a question asked in once in NEET exams, which is not the characteristic feature for being the genetic material. So they gave an answer, it should be unstable. So your answer will be unstable, क्योंकि genetic material को हमेशा stable होना चाहिए. Okay. RNA जेनेटिक मटेरियल है कौन कौन से प्लांट में, दैट इज टोबैको मोज़ेक वायरस, क्यूबी, बैक्टीरियोफाज. तो दीज आर द एग्जांपल्स दे माइट आस्क इन योर नीट एग्जाम्स. ओके. स्टेबिलिटी एज वन ऑफ द प्रॉपर्टीज ऑफ जेनेटिक मटेरियल वाज वेरी एविडेंट इन ग्रिफिथ्स एक्सपेरिमेंट, क्योंकि जब उन्होंने हीट किया था बैक्टीरिया को, देन इट कुड नॉट लाइक ट्रांसफॉर्म द विरुलेंस ऑफ द बैक्टीरिया. ओके. तो इससे हमको पता चलता है कि जेनेटिक मटेरियल बहुत हीट स्टेबल होते हैं. ओके. Reason why DNA is genetic material, not RNA. What is the reason? O' hydroxyl group present at every nucleotide in the RNA, that it is very reactive group, okay, and makes RNA labile and easily degradable. So RNA is also now known to be catalytic and hence reactive. तो इसीलिए DNA होता है genetic material, RNA नहीं होता. And the final one, दैट इज डीएनए आरएनए दोनों ही म्यूटेट कर सकते हैं, बट आरएनए इज अनस्टेबल, तो इसमें म्यूटेशन ज्यादा होता है, इसीलिए वायरस का ज्यादा पैंडेमिक देखने को मिलता है, क्योंकि दे गेट म्यूटेटेड एंड वी डोंट हैव ट्रीटमेंट एंड एंटीबॉडीज फॉर दैट. ओके. सो दिस इज द फर्स्ट वीडियो मॉड्यूल that contained the mainly structure of DNA, how we proved that the DNA is the genetic material. Okay, so this was the content of the first video. In the next video module, we will be discussing about the replication. Okay, thank you.